Oh, got it. Okay. Thanks, Philip, for the nice introduction. Makes me blush a little. And thank you all for inviting me to your seminar series. It's a real pleasure to be here. I hope you like what you see. So I joined McGill maybe about 4 years ago. And so the talks these days are still a little uneven. And what you'll see today is a little bit of some older work that I did in my postdoc advisor's lab, in Josh's lab, mixed with some unpublished stuff from my lab and a recently published paper that we had this fall. So our general interest is to learn more about how the brain becomes wired and how this wiring, which is really the connections among neurons called circuits, give rise to a wide range of our abilities. So things that span from as simple as reflexes through sensation and motor control, perhaps all the way to more cognitive kinds of things. Now, the connections themselves are set down earlier in development, and they seem to revolve around the choices that neurons make, choices about whom they should connect with. Now, the manner in which they go about making these decisions, the factors needed to bring many neurons together, and indeed the consequences of some of these early wiring events on overall function, and as happens all too often, dysfunction, are truly major open issues in the field and are issues that really motivate our work. So we try to learn more about this problem by studying the neural circuits located in the earliest step of the visual pathway of mice. And this is the mouse retina and its immediate targets within the brain. The reasons for retina will become clear in a moment, but suffice to say it offers us a bunch of advantages. The first advantage is going to be really familiar. It's a piece of the CNS, but it's located outside the skull. It's in fact a piece of the diencephalon that, can I go forward here? Yeah, that gets pushed through the skull to form a thin sheet of neural tissue located on the back of the eye, onto which images of the world are focused by a lens, also within the eye. Now, if I were to take a cross-section through this structure and show you a cartoonified version of this cross-section, what you'd see is a really pleasing form of regularity. So all the neurons here live in 3 layers. There's an outer layer containing the photoreceptors, an inner one containing the inner neurons, and a ganglion cell layer containing the so-named cells, whose axons bundle together to form the optic nerve and convey visual information off to the brain for further analysis. Now, some 60 years of truly elegant studies has taught us an enormous amount about the circuits within this organ and how they might be arranged and how they might work together to support vision. So to just pull out a few facts that are germane to the talk, we know that every single one of these circuits begins with a photoreceptor. These guys are photon detectors. They're like the pixels in your phone cameras. Every single one of these circuits ends with a retinal ganglion cell, which, unlike the photoreceptors, come in a variety of flavors or cell types. And in mice anyway, each of these types appears to be specialized to detect a particular aspect of the visual scene, such as edges that move in a particular direction, those perhaps with a certain orientation, patterns, colors, contrast, so on and so forth. Now, each of the ganglion cells detects their visual feature completely independent of their neighbors. Okay, so the pink guy doesn't know that the other cells exist, but he arranges himself across the retina in parallel. And so what this means is if I were to label this pink neuron across the retinal surface, and on the next slide show you its distribution on a real retina, what you'd see is that it's replicated itself all across the retinal surface. So here's a retina, it's been unfolded in a mouse line in which we've labeled that ganglion cell. You can kind of see some axons streaming off to this PowerPoint disk, which covers the optic nerve head. And what you can't appreciate here is that types like this, pure types like this, arrange themselves in an almost hexagonal-like lattice, okay? And it's called a mosaic. Now, all the ganglion cells are doing this. It's not just the pink one. And what this means is there's a growing sense that the eye isn't just a camera. The retina doesn't just take a picture of the world and send it upstairs to the brain, but rather it might work as sort of a compressed sensor and decompose an incoming image like this into a series of images, each of them containing emphasis on a certain aspect of this painting, which in turn reflects the feature preference of a given ganglion cell over here. Now, what the brain does with a signal like this, whether it flattens it back up and creates the Mona Lisa again, or combines it with, certain concepts, or yanks out images from this deck to drive specific behaviors, is a fascinating question. There's some folks in the lab who are working actively on that. But for today's talk, and I think for the bulk of my work on the retina, what we're going to focus on is the first mystery, which is how on earth these neurons even acquire these remarkable preferences. And so, without going into too much detail, we know that it's nothing special about these ganglion cells. There's nothing intrinsic to these neurons that should let them be able to perform this task. Instead, the general consensus is that each ganglion cell is endowed its feature preference by synapses it receives from a very specific mixture of interneuron types, of which there's about 120 or so. And I should probably update the slide. This is closer to 50 now. So the specific questions in my lab are things like, how does the pink ganglion cell find and wire specifically to the pink interneuron over here? What are the factors that direct that match? And given that they spend so much energy to connect to one another, what is the contribution of this interneuron to this ganglion cell's feature preference? These are the kinds of things that we want to learn more about over the next few years. And in the next few slides, I'm going to share with you some of what we've learned in each of these three categories. But before getting there, I need to kind of step back a little and tell you about the genetic approach we take to addressing these kinds of questions. So if we wanted to learn how purple finds purple, or how green avoids pink, or what the contribution of yellow is to the yellow ganglion cell's receptive field, or its feature preference rather, we'd ideally want to turn the retina into this cartoon, right? That is, we'd want a way to be able to mark and manipulate every single one of the neural elements in here. And without going into tons of detail, it turns out that a concerted effort by big consortiums like GenSat, the Allen Brain Institute, and smaller labs like my own, and like the one I postdoced in, have led to an encyclopedia of mouse lines that allow one to do just that. And on the next slide, I'm going to show you some retinal cross-sections from mouse lines we have in the lab that label specific types, with interneuron lines up top, okay, and ganglion cell lines down at the bottom. Now, there's a couple of things I'd like you to notice about these images, in addition to them being pretty, or at least I think so. The first is how stereotyped the retina is. You see these red lines that run through each of these images. These are the processes of an amacrine cell called a Starburst amacrine cell. And we label it using antibodies to an enzyme called choline acetyltransferase. What it does for us is it divides up the specialized retinal neuropil, which contains all the circuitry of the retinal circuits, into 5 layers. So 1, 2, 3, 4, 5. Same up here. 1, 2, 3, 4, 5. The other thing I'd like you to notice is that each of these retinal types appears to confine its axons or its dendrites to just 1 or perhaps 2 of the 5 possible alternatives. An arrangement that led a fellow postdoc in Josh's lab, Xin Duan, and I to wonder, well, maybe the way that you get cells like this to wire up specifically to cells like that is because they know which of the 5 alternatives in which to grow their dendrites and axons come into synaptic striking distance of one another, connect and form circuits. And so to investigate this idea further, what we did was to get a hold of lines like this and seek out expression of recognition molecules, more commonly known as cell-cell adhesion molecules, that would be expressed in cell type specific patterns that might indicate a role in guiding neurons to these kinds of locations. And what we learned was that these 2 neurons here, these bipolar cells, they have names, bipolar 2, which projects to layer 2, and bipolar 5, which projects to layer 4, expressed a pair of highly related cadherin molecules, with CAD8 labeling type 2 bipolar cells and CAD9 labeling type 5s. Now, given that these were the only 2 neurons in the retina that expressed CAD8 and 9, and given the famous roles of cadherins all over the nervous system during neural development, we wondered whether these proteins were being used by these cells to find these locations, enter into these layers, and wire up to targets. And so to test this idea, what we did was to get a hold of Cre lines that granted genetic access to these 2 cells, use Cre-dependent viruses containing GFP to reveal their anatomy, and then we studied their anatomy in the presence and absence of cadherin. And what we found was something surprising. So when we looked at type 2 cells in the absence of their cadherin, we found that approximately half of these cells stayed in the home layer, layer 2, and half of them moved off to a location that approximated where the type 5s live. Deleting a cadherin this neuron doesn't express has no obvious effect. Doing the same experiment with the type 5s revealed a complementary deficit. Half of these neurons stayed in their home layer, and half of them moved off to a location that approximated where the type 2s live. These two neurons are more related than we initially thought. They seem to be making a decision for two mirror-symmetric positions in the inner plexiform layer, a decision in this neuron that seems to require CAD8 and places it in the top layer, and a decision in this neuron which requires CAD9 and places it in layer 4. Now, given this requirement, we immediately wondered whether the CADs are telling them where to go. And so what we did was to get a hold of our Cree lines, our Cree-dependent reporters, and then we swapped their cadherins. And it turns out that it doesn't matter what bipolar type you are. What matters is what cadherin you express, because we could take a type 2, give it CAD9 and push it down and make it look like a type 5, or we could take a type 5 bipolar cell, give it CAD8 and push it up and make it look like the type 2 cell. The two cadherins impart positional information to these two bipolar cells, bringing them to either layers 2 or layer 4, presumably so these cells could wire up with postsynaptic targets and create the circuits that we started off talking about. Who would those targets even be? Well, as it turns out, we had a really great clue. You see the red lines that populate each of these images, the processes of this starburst amacrine cell. This is a rather famous neuron in the retinal literature because it provides direction-selective inhibition to a ganglion cell called a direction-selective ganglion cell. And it has dendrites that go exactly to these two locations, layers 2 and layers 4. Now, given that we have 4 kinds of cells that target 2 layers, and we know why 2 of these neurons show up there in the first place, we immediately wondered whether these neurons form a circuit together. And so to investigate this idea, and indeed to have a somewhat more general strategy to map connections between neurons in the retina, I devised the following trick. We take mice that express GFP in particular ganglion cells like this. We then cross them off to ones that put the enzyme Cre recombinase in interneurons like that. Pretend it's NETO1 Cre. Then, either by way of a virus or by way of a mouse cross, we selectively install the light-sensitive ion channel rhodopsin 2 into the interneurons membranes, record from the ganglion cell using whole cell electrophysiology, whole cell patch clamp, and then we activate the channel rhodopsin on these interneuron membranes using 2-photon excitation. And the reasons for this I could get into, but briefly, we can't use visible light in the retina. The photoreceptors live up here and would see it. And the properties of 2P, creating a small spot, kind of like my fake laser pointer, offered the kind of spatial confinement that made this experiment possible. And I should just say that at the time that I did these experiments, it was tough to make a commercial scope do this kind of trick. And so I was quite fortunate to learn from Ed Susi and Joel Greenwood and others at Harvard how to build these kind of scopes. And I'm happy to talk more about these details afterwards if you're curious. So what we'd like to do is what I'd like to do is to use this tool now. Let's use it and do an experiment and see the kind of functional connectivity measurements we get out of it. So on the next slide, I'm going to show you the position of a ganglion cell I'm recording from and a field of bipolar cells that I'm stimulating. You won't see the real neurons. They live in 2 different layers. The recordings are done in flat mount, unlike all the cross sections I've shown you so far. So here's the ganglion cell. Here are the positions of 200 or so bipolar cells, type 2 bipolar cells. We're going to stimulate each one using our 2-photon trick, let's say 10 times, signal average on the green cell, and then I'm going to place the current we measure back in the location of the stimulated neuron. And when we do this across the field, we end up getting all of these beautiful heat maps. And so you can see that this neuron receives most of its bipolar input from the south side of the soma. And I've colorized each of these currents according to strength. And for those of you in the know, the electrophysiologists in the audience, these are inward currents recorded at -60. They're blockable by glutamatergic blockers, exactly what you expect for a bipolar RGC synapse. The tilt that we observe here is also quite gave us a lot of comfort because we knew that the ganglion cell we were recording from grows dendrites with a pronounced tilt. So now we have a way of mapping connections between inner neurons and ganglion cells. So what about our original question? What about the four neurons? Do they connect with one another? Well, you're already looking at the type 2 cell, so they form excitatory connections. The type 5 cell connects with the direction selective cell 2. We confirmed that the type 5 cell provides excitatory input to starburst amacrine cells. And we also confirmed an older result showing that the starburst amacrines provide inhibition to the ganglion cell. The four neurons have this particular synaptic relationship with one another. And we know why two of them show up here in the first place. So what's going to happen? What's going to happen when we go in and delete Cat8 or 9 from these cells and redo our connectivity measurements? It's not entirely clear, right? Because if you remember the phenotype, when we delete Cat8 from here, approximately half the neurons stay in the home layer and half of them move off to a location that approximates where the type 5s go, right? Seemingly well within synaptic striking distance of this bottom dendritic arbor, maybe nothing would happen. And so you can imagine our surprise when we went in and did the experiment and we found this massive reduction in connectivity. Okay, so normally within the dendritic arbor of these direction selective neurons, somewhere in the ballpark of about a half of bipolar cells above those dendrites connect with the ganglion cell, but in the absence of Cat8 and 9, somewhere in the range of 10% are now connected, a dramatic loss of connectivity. And I simply don't have time to talk to you about the consequences of these deficits for the visual function of this RGC, but suffice to say that this neuron likes to sense the motion of an edge being a place in the visual field that goes from dark to bright or bright to dark. And what I haven't told you is that bipolar 2 is an off bipolar cell and would convey those dark signals, and bipolar 5 is an on bipolar cell and would convey the kind of bright signals. And I'm happy to talk to you more about those studies afterwards if you're curious. What I'd like to do instead right now is to kind of tell you a bit about how this story has evolved since, gosh, 6 years ago, 7 years ago now. At the end of the story, we were feeling pretty good, right? We were feeling pretty good about this direction selective circuit. So we knew about these four neurons. We had independent genetic access to each of these different types. And we had sort of a partial list of wiring instructions, right, that would set up this kind of circuitry. But there was always something that bothered us about this experiment, right? Cadherins like to bind to themselves across cell-cell junctions, right? The idea being that this guy should bind to itself on some target in layer 2. How do these CADs bring these neurons into these layers? How do they position the axons into these locations? We didn't really know that. And so it would turn out that a couple of ongoing efforts in the lab, in Josh's lab, would help us out. The first was the observation that cadherins 6 and 10 pattern the postsynaptic neurons. And the other was an older observation from the literature, which showed that cadherins like 9 could in fact bind to other CADs, so they could bind heterophilically in certain conditions, often binding to their close relatives. It turns out that the closest relative of 9 is 6 and 10. And so what Shin did was to generate triple knockouts in which we could ask, well, what happens if we remove these postsynaptic cadherins or remove all 3 cadherins? What would happen to the laminar position of these various neurons in the circuit? It turns out to be a difficult thing to do. He had to use CRISPR-based genome engineering to get it done. And that's because these genes are back-to-back on chromosome 15. And I'd like to share with you a couple of images from this study down here, in which we analyzed these triple knockouts and looked at the ganglion cells and found that in the absence of 6, 9, and 10, the ganglion cells now lose their way. So in controls, they form these thin stratified dendritic arbors. In the absence of 6, 9, and 10, you can see they elaborate these kind of more disorganized, scrambled looking structures. But interestingly, you'll notice that the starbursts don't care, right? In the absence of 6, the starbursts are perfectly fine. And what I haven't told you is that these neurons are amongst the first neurons in the retina to grow these stratified kind of layer-like dendritic arbors. Could these cadherins work as sort of an overlapping kind of adhesive framework, kind of an adhesive scaffold? Well, to address this idea, what we did was to get a hold of this tree line down here install a receptor for diphtheria toxin into the starburst, kill the starburst early in development, and then examine the lamination of these RGCs. And we've found phenotypes that were remarkably similar to the case where we deleted the cadherins. It's almost as if in the absence of 6, 9, and 10, the ganglion cells just can't see these two locations anymore. And I'm happy to talk more about this afterwards if you're curious. What I'd like to do right now is to kind of zoom out a little bit. Could it be the case that every layer in the retina gets a cadherin? Seems a little silly, but from wiring point of view makes good sense, right? It keeps appropriate synaptic matches in close proximity, keeps inappropriate matches far apart. It turns out that our preliminary list of cadherins within these layers isn't too far off of this simplistic diagram. So we do have some candidates in the other layers, and I'd like to share with you some results that a student in the lab, a PhD student in the lab, Aline Rangel-Olguin, has been getting with isoform 4, otherwise known as retina cadherin. So Aline has been investigating this isoform and its role in setting up circuitry in the retina. And one of the first experiments she did was to get a hold of antibodies to CAD4 and stain retinal cross-sections with them to just see where this protein is distributed. When she did this, she found a pair of bands kind of in the upper part of sublamina 3 and into sublamina 1, okay? Then by getting hold of a mouse line in which the cadherin 4 gene has been interrupted by the presence of a Cre-ER fusion and crossing those heterozygotes off to ones that contain a Cre-dependent reporter in their genome, treating with tamoxifen, Aline was able to figure out what the cells are that would be expressing this protein over here. And by and large, these turn out to be ganglion cells with dendrites positioned within the upper part of sublamina 3 or into sublamina 1. Now, there was another cute aspect to these results that we found, which is that actually CAD4 labels an enormous number of ganglion cells, about 600 to 800 cells per millimeter squared in some of our high density tamoxifen injections. This is about a third of all ganglion cells. And what I haven't told you is that this lamination pattern corresponds to the great division in the retina between off ganglion cells and on ganglion cells. Retinal ganglion cells only coming in about 3 flavors, right? On, off, on, and off, and we're labeling about a third. Could CAD4 help the off ganglion cells climb into these upper layers in the IPL, collect input from off bipolar cells, and then become functional? This is the hypothesis that she's been testing over the past couple of years or so. And some of her first experiments were functional. So what she did was to cross the CAD4 gene into a separate line in which we have genetic access to all ganglion cells, deliver the genetically encoded calcium indicator, GCAMP6F, into these retinae, image multiple locations in the retina. This is what a single field would look like. Extract the neural responses from these various neurons over here using a machine classifier called Sweet2P. And this is what the traces would look like. So we're looking at every ganglion cell in a control retina. And so here's a stimulus, it's a full field flash. It goes from gray to dark to bright to dark to gray. You can see on cells here. You can see off cells. If we squint somewhere, I'm sure we can find on-off cells, probably around here. And so she collects all this data. Now she's collecting thousands of neurons worth of data, so I can't show you the traces like this on the next slide. So instead, I'm going to turn this into a picture on the next slide, and I'm going to sort the data by placing all the off cells in one part of the picture and all the on cells in the other part. So just follow my pointer. Here we have the same data where each column is a cell's response, and the hotness of the color reflects the strength of the signal. And we have gray to off to on to off gray. And over here, the same thing, right? So the off cells are over here, the on cells are over here, and this kind of railroad track pattern in the center tends to be these transient responses we get from on-off cells. And we do this by computing an on-off index, which is quite simple. It's just the value at on minus off divided by their sum. Doing this gives us a histogram, right? This is a histogram of the same data up here, and you can see the same results, off cells, on-off cells, on cells. And so what's going to happen? What's going to happen when we delete CAD4? Well, if this CAD works the same way as the other bunch of CADs do, then the off ganglion cells might lose their way, right? And at least deplete from this off column. In fact, what we see is almost a switch. So we deplete from the offs and we sort of gain cells that are responsive to light onset, almost as if the ganglion cells that lose their way in CAD4 maybe are picking up inappropriate input from on bipolar cells. And in a limited number of single cell labeling experiments, that's what Aline finds. So in heterozygotes, ganglion cells in CAD4 tend to project into these upper laminae, the off laminae we've been talking about. But in the absence of CAD4, they end up having this scrambled look that is so characteristic of these cadherin mutations, oftentimes with cells growing ectopic dendrites into on laminae. And I'm happy to talk more about what we're doing with this story and some overexpression type ideas we have and stuff like that afterwards, if you like. What I'd like to do right now is to pivot sort of into the last-ish part of the talk and just consider this cadherin idea a little more. So we have this notion that cadherins might place neurons within the particular layers, and that makes good sense because you bring appropriate matches within close physical proximity, keep inappropriate matches far apart. The problem is we have 45 different types of ganglion cells, nearly 50, 120 different types of interneurons, and only 5 layers in which to shove all of this circuitry. It's got to be the case that there are functionally different kinds of ganglion cells, functionally different circuits that occupy the same physical lamina. If so, how does a neuron like this know to keep its wiring apart from this interneuron over here? How does it make this kind of choice over here? This is a difficult problem, right? And I think it's well illustrated by some reconstructions that are coming out of serial section EM work. I'll show you one on the next slide. I don't think it's from retina. I think it's from somewhere in cortex, but retina would look very similar. So here's a cube of some, I don't know, call it 50 microns on a side. Each of the processes within this cube is colored according to a different neuron, and you can see that it's a tangled mess, right? So how do neurons grow into this area and synapse with the correct target without getting fooled and inappropriately synapsing with all of these equally proximate alternatives? And so we learned something about this when I was postdocing with in Josh's lab by investigating another recognition molecule called sidekick2. So it looks like this. It has Ig domains in its ectodomains, 6 of them, belongs to the immunoglobulin superfamily, contains a PDZ binding motif, which puts it right at the business end of this kind of specificity that we're talking about, forming connections. And what I discovered was that a pair, an interneuron RGC pair, express sidekick2 selectively. They look like this. So one is called a VG3AC because it expresses VGLU3. Another one is a W3B RGC. And in experiments where I was mapping connectivity between these two kinds of neurons, what I found was that connections between them were incredibly strong and incredibly faithful. So anytime these two neurons overlapped, there was a connection. But in the absence of sidekick, this strong connectivity was reduced, right? So in controls, somewhere in the ballpark of, I don't know, nearly 100% of presynaptic neurons will connect with this ganglion cell within about 100 microns, which is the dendritic capture of this ganglion cell. But in the absence of sidekick2, we end up reducing this connectivity strength. Fewer cells are connecting, and those that were connected were often exhibited weaker currents, weaker functional synapses. And so this idea makes you think that, well, maybe this is the role of proteins that belong to this Ig superfamily. Maybe they mediate sort of a local biasing of connectivity between proximate partners and help keep the connections of a neuron like this and a neuron like that straight. And so to investigate this idea further, what we felt like we needed was to generalize this result. And that was the job of my PhD student, Pierre-Luc Rochon, who teamed up with Aline from the CAD4 project and a fellow graduate student, Catherine Theriot, in the lab. And what Pierre did was to focus on the closest cousin of sidekick2 called sidekick1. And I'd like to share with you some of his results. You know, my timer is off because we spoke in the beginning, but I'm going to just pretend that I'm on time. Maybe
You're all good. You're all good.
I'm good. All right, good. All right. So let's get into some of Pierre's data on Sidekick 1. So one of the first experiments he did, it's actually the sort of theme of investigations in my lab, is to get a hold of some mice in which the Sidekick1 gene has been interrupted by a Cre-GFP fusion. He then took retinal cross-sections and retinal whole mounts from this line, stained them with GFP to see which cells express Sidekick, and with markers that labeled bipolar cells, amacrine cells, and ganglion cells. And so that's shown here, cross-sections, whole mounts, bipolars, amacrines, ganglions, bipolars, amacrines, ganglions, and some counts over here. The main takeaway from this is it's an awful lot of cells. And initially we were a little dismayed because how are we going to choose, right? How are we going to focus on one of these ganglion cells and its potential interneuron partner? Seemed like a tough proposition. So what Pierre and Aline did was to team up and analyze publicly available single-cell sequencing atlases held at either the Broad Institute or there's a couple that have just been published. And they searched out Sidekick1 expression within ganglion cells, and they found that, in fact, Sidekick really only labels 5 kinds of ganglion cells. 2 of them are known, on alpha cells that respond to light onset with sustained responses, and type 2 melanopsin-positive RGCs. 3 of these RGCs are predicted from the sequencing data, but had no real-world counterpart. Now, by identifying these cells, they then sought out type-specific markers that could identify them along with staining with GFP. And so that's what Pierre did. He took antibodies to these various proteins, and he stained retinae like this, and then he sought out the kinds of spaced-out mosaic-like patterns that characterize a pure cell type. And he turned to the alpha and M2 first to validate his approach. And so here is a retinal whole mount stained for osteopontin and neurofilament heavy. The encircled cells are the triple labeled neurons. Those are the on-alpha cells. This guy over here. Pierre then measured distances between these neurons and computed what's called a density recovery profile. It's just fancy for saying that the spacing between neurons of this kind is somewhere on average, like, I don't know, 75 microns or so, and the total density of this neuron is low. Using a related strain in which the Sidekick1 gene has been interrupted with a Cre-ER fusion, let Pierre label this kind of cell sparsely and identify it using stains like this. Turns out to look exactly what one would expect for an alpha cell, large dendritic arbors located in sublamina 5. And I'm just not going to show you the M2 RGC for some technical reasons we could talk about afterwards. Next, Pierre focused on the brain3C RGC, labeled by this guy over here, and found a relatively more dense kind of ganglion cell that grows dendrites into the middle sublamina of the retina. And then the final pair of RGCs, labeled by a transcription factor called NR2F2 and calbindin, turned out to be RGCs that grew sort of medium field dendrites, often showing a windswept appearance that targeted sublamina 4. Now, with markers like this in hand, Pierre figured, well, what we should do is do Aline's experiments and GCamp image from retina like this and stain them with markers afterward to associate these kinds of morphological parameters to their functional preferences. And so the way he does this is to take the 2-photon image from many locations, label the blood vessels using a dye, permanently label those blood vessels after imaging with a dye with a lectin, register the 2 images together, and then he wrote some code to do landmark correspondence between the 2-photon and 1-photon images that lets him refine every single one of these fields within the post-stained version of themselves. And here's the blood vessels, here are the GCamp positive cells. He highlights ROIs, collects marker labeling from within the marker stain channel, and then can extract the responses, in this case of the brain3C RGC over here, okay? So doing this for the whole family of ganglion cells we've talked about, starting with the alpha cells, reveals the expected results. So Pierre clustered the on-alpha and the M2 together using osteopontin, and that's just because of antibody restrictions and for some other reasons we could chat about later, but gives the expected kind of visual response, sustained sluggish responses to light onset, the same kind of response to a bright bar moving in 8 different directions with no obvious preference for bar direction. Doing so on the 2 NR2F2 positive ganglion cells revealed acute surprise. It turns out these are a pair of on-direction selective neurons, with the NR2F2 calbindin positive guys labeling neurons that detect motion ventrally, and the NR2F2 alone guys detecting motion dorsally. Right? The brain3C RGC turned out to be an on-off cell. Actually, it had sort of an interesting property. So when we flashed the entire field on and off, this neuron appears as an on RGC, but when we use a bar traveling in different directions, we often saw transient responses to the leading and trailing edge of the bar. And when we plotted these data as a polar plot, they often showed this kind of elongated look to them, suggesting that maybe these neurons like axial motion or sort of edges of sorts. And some of this, some of the extra details are quantified over here. We could talk about that afterwards if you wanted. What we really wanted to do was, with this data in hand, ask, well, is Sidekick-1 helping these kinds of circuits get wired, right? Where would we expect phenotypes to appear? Well, as it turns out, some parallel studies in the lab analyzing the sequencing atlases, as well as some sequencing work we had done, showed that these 3 neurons up here are actually Sidekick-1, Sidekick-2 double positive neurons, whereas this brain3C RGC expresses Sidekick-1 alone. And so we named it the S1 RGC, and we decided to focus on this guy. So when Pierre did his experiments, he simply repeated this kind of experiment in the knockout. He found more or less the expected result, which is the deficits, the phenotypes, were found mostly on the brain3C RGCs and led to a loss of responsiveness on this neuron. We did observe subtle deficits on the direction selective neurons, oftentimes inappropriately pointing direction selective vectors. We could talk about why I'm not going to talk about these guys in the next few slides, if you like. We can talk about that afterwards, if you like. But for the remainder of the study, what we did was to focus on these brain3C RGCs and the on-alpha RGCs, which showed no obvious deficit. And the reason for this is because it turns out we can selectively label these 2 neurons and target them with patch electrodes using retrograde injections of virus in either the colliculus or LGN. Okay, so here we deliver retrogradely infecting AAVs containing a Cree-dependent reporter into our Sidekick mice. Here's a 2-photon field taken from a retina just like this. It shows an on-alpha cell and an S1 RGC. We target them with electrodes and we show moving bars to these 2 neurons, and we get responses that look very similar to what we saw with calcium imaging. So on-alphas don't really care about bar direction. S1 RGCs often show this teased out axial kind of shape. And so with this kind of labeling method in hand, we then focused on these 2 neurons and characterized their receptive field, starting first with a small spot positioned over these 2 neurons that grew subsequently, okay? And what we found was that the S1 RGC turns out to be an on-off cell when the spots are small, but then the off responses become totally inhibited when the spots are relatively large. The on-alpha also showed some suppression, but not nearly as much, matching previous reports about its response profile. Now, these kinds of polar plots made us feel like we needed a different kind of stimulus. And so what we invented was a bar centered over the receptive field of this neuron, a stationary bar that rotated through many different kinds of angles. Presenting that to these 2 neurons, again, sort of confirm the shape of these 2 things over here, with the S1 RGCs often having a preferred bar orientation and firing very little to bars oriented orthogonally, the on-alpha cells not really caring about bar orientation at all. And so what's going to happen when we go in and do our knockouts? We already have a feeling that something should be up from our calcium imaging. And so when we went in and did the same experiment in our Sidekick knockouts, we found a fairly dramatic loss of responsivity on the S1 RGCs, with off responses being almost totally absent. The on responses were often weak or disorganized feeling. Similar effects down here for the oriented bar, but to some degree that could be inferred from the stimulus over here. And this is kind of quantified by some graphs over here, which I'm happy to talk about. So given this kind of phenotype, we really wondered, well, maybe Sidekick is matching the S1-RGC up with Sidekick positive interneurons. And that matching process leads to specific connections and leads to responsiveness on these particular neurons over here. So to investigate this idea further, we simply repeated this exact same experiment, but went whole cell to look at the excitatory and inhibitory synaptic currents on these ganglion cells to a small flashing spot that grows sequentially over time. The next slide is a little busy, but just follow my pointer. I'll take you through it. So in the top part, we're looking at inhibition, inhibitory currents, and we're isolating them by holding the cell at 0 millivolts. Down here is excitation. These two are controls, all right? So you can see that inhibition grows as the spot size increases, right? As you recruit more of the surround. The excitation peaks at a certain point, probably the receptive field center size, and then decays, probably due to some presynaptic inhibition that's being recruited. But in the absence of Sidekick 1, we end up strongly reducing both the inhibitory and the excitatory synaptic currents, suggesting a loss of functional synapses on these neurons over here. Now, for completeness sake, we also did the same experiment using our oriented bar stimuli, and we found kind of a cute result, a resultlet, if you wish, which is that bars that produce the strongest excitation were often oriented 90 degrees away from the bars that produce the strongest inhibition. And that was true regardless of whether we use dark bars or bright bars. And we kind of wonder whether it's this sort of bias, it's a little subtle, but we wonder if this kind of bias over here is what leads these S1-RGCs to have their kind of preferences for edge orientation, if you wish. Now, doing this same experiment in the knockout also shows the expected effect from the spot experiment. So strongly attenuated synaptic currents, suggesting a loss of synapses in the absence of Sidekick. Now, I should just say that this kind of receptive field arrangement has been seen before, so it's out there in the literature. In fact, there's some really nice work from Greg Schwartz's lab describing a different pair of cells. But over here, you might imagine a receptive field kind of structure where you have an elongated center shape, so that if your bar is oriented like this, you get strong excitation, whereas if a bar is oriented like this, you get strong inhibition. And I should just say that there's older work in the literature from other species that suggests that an amacrine cell that kind of looks like this, a very wide field amacrine cell, is the one that's providing this kind of tuned input, right? This kind of oriented tuned input. And I should just say that we found an amacrine cell that looks awful similar to this neuron that is Sidekick 1 positive, goes to sublamina 3, and overlaps the S1-RGC pretty well. And so we're actively pursuing the idea that maybe these inhibitory currents arise within this neuron. We could talk more about that afterwards if you like. So what is the reasoning behind this loss of functional synapses? Maybe the Sidekick works like the cadherents. Maybe these neurons just can't find sublamina 3. We don't think that's true because at least at a gross level, the two neurons do target the same layer in the inner plexiform layer. But however, when we reconstructed the dendrites of these two neurons, we did find dendritic deficits. So in fact, the Sidekick negative cells did have dendritic arbors that were slightly shrunken or less complex, I suppose would be the more correct way of describing them, with fewer intersections in a Schall analysis and less overall length. And so whether this dendritic effect accounts entirely for the synaptic deficits we've seen before, or whether this is the result of dendrites just not receiving enough synapses, is a thing that we're actively pursuing in the lab. And I'm happy to talk more about that afterwards if you're curious. I think I should start rounding it up. I want to just point out that we have a really great understanding of sidekicks in the retina and the family of circuits that these two proteins label. The two that really interest us are the W3B VG3 cell, which I investigated as a postdoc, Sidekick 2 positive, and the S1-RGC and its potential binding partner, its potential synaptic partner, the CA2AC, which are both Sidekick 1 positive. These two neurons have different feature selectivities. They target the same layer, right? They're functionally distinct, but are physically entangled. Could it be that the way that these two neurons set up their connectivity, despite being stuck together, is because of this choice of sidekick 1 and 2? If true, then if we were to swap sidekicks, then we ought to change wiring. And that's something that we're trying to do in the lab. And I could talk more about those experiments afterwards, if you like. I think at this point, I'm going to pause. I'm going to sort of pause and summarize what I've told you. So we began by wondering how circuits assemble. And in the retina, we know that they assemble within this specialized region called the inner plexiform layer. It's divided into 5 sublayers. Factors that are beyond the scope of this talk set up those layering patterns, but once set up, proteins like cadherins help neurons find a particular location within this plexiform layer and come into proximity with potential synaptic targets. Now, this mechanism, although great, can't account for all the specificity that these kinds of cells need, because once within these layers, they encounter a number of proximate partners, and they have to decide who to synapse with. And we believe proteins like sidekick are important for that kind of synaptic specifying effect. And I didn't talk about it a ton today, but I sort of alluded to the consequences of these types of wiring mechanisms on object motion and motion direction. And I showed you some data for the S1 RGC in the sensation of edges or edge orientation. And I think with that, I should stop and thank everyone who helped. So some of the work you saw today was done in my advisor's lab, Josh Sains, with a lot of advice from a co-mentor of sorts, Marcus Meister. The cadherin and sidekick 2 stories were done in collaboration with my friends Shin and Masa in Josh's lab. I showed you the work of 2 excellent graduate students, Pierre-Luc Rochon for sidekick 1 and Aline Rangel-Olguin working on cadherin 4 with help from Catherine Theriot in both cases. And I put up this sort of Cajal drawing of the early visual pathway to remind myself to mention that about half the lab is interested in exporting the strategy of retina, right? This link between connectivity, wiring organizers, and function into some subsequent stages. And we're pursuing this as collaborations with Eric and Anmar in my department. And I should thank funding and all of you for your attention. Thanks very much.
Thank you very much, Arjun, for this absolutely fantastic talk. I hope we'll have plenty of questions. So
it's a little dense, but yeah. Yeah, somebody named James is raising his hand.
Yeah, James has a question. James, go ahead.
Hi, Arjun. Thanks for that spectacular talk. It's very interesting stuff, and the techniques that you're using are, very precise and something that I hope to, maybe apply in my own research moving forward. I was wondering about the lamination of those that inner plexiform layer that you were showing, and most of the connections that you showed, or all of the connections that you showed, were relegated to just one layer. Are there types of cells that might connect to 2, 3, 4, 5, multiple layers? And, is that part of this information processing?
Yeah, so there are cells that span the layers. In the retina, I guess I think about glycinergic amacrine cells often have a narrow shape and extend their dendrites throughout the 5 layers. I should just say the 5-layer thing is kind of, I inherited it from Josh's lab. Some people define it as 10 layers. Doesn't really matter, okay? There's many layers in there. These kinds of amacrines do extend across many layers, but it seems as though, although they look that way, they receive inputs in a particular layer and then often have their outputs in a different layer. Yeah?
Oh, interesting. Okay. Yeah. Thank you.
And whether or not these kinds of cells, which grow whatever translaminarly, just make up a word, whether those cells have some different strategy for finding targets, unclear. I don't know. You might think so, right? Given that they're not confined to a given layer. Let's see.
Yeah. Well, it makes me think of just, the kind of information that you might get from combining layers together. But yeah, thank you.
All right, Philippe has a question.
Yeah, I was looking at your presentation. It reminded me that I like studying the eye because it's a simple organ. Well, obviously not, but alpha RGCs, I mean, I just remind, they are responsible for the looming effect, if I'm right, huh?
Because so one of them there are so in other species, I think there are 4 types of alpha cells. They're sustained and transient, on and off, and do the combinations. And I think the one that you're thinking about in mouse is the off transient RGC. I'm trying to remember the paper. It's a paper from the Rosca group. And it does in fact have looming responses, retina in a dish. I don't know whether anyone's targeted that cell and seen whether it does the same thing in vivo or not, though. But you could.
Okay. Yeah. Thanks.
Jeremy?
Thank you so much. That was a really lovely talk, and it was just kind of building on what Philippe said. I don't know if the retina is simple, but you did a really nice job of taking very complicated ideas and sort of simplifying them down. But now I'm going to ask a question to try and complicate it again.
Okay.
Which is while you were, I hope I can articulate it well. While you were talking, is particularly the sidekick story, and sort of discussing, I think by necessity, these interactions or these circuits in terms of almost sort of columns within the retina. So like bipolar, I suppose, and amacrine and ganglion cells, and what's defining those connections, and then this sort of bar angle versus, I guess, traditional spot on-off ganglion cells that we have orientations of bars. And it made me think about lateral inhibition or lateral enhancement, like lateral interactions. And I'm just wondering if you sort of thought about that, and if that's possible to add in, or if that just kind of blows the complexity up, a huge amount, to sort of start to think about how those columns would impact each other depending on which cells are next to each other.
Yeah, I think I know what you mean. So it's sort of a question about, I kind of draw the circuit as like, here's the only circuit, right? But of course, there's so many other ones nearby, right? And you're saying, could the interneurons responsible for one influence another, right? Or could signals from one influence another? Well, we know that's probably not true at the ganglion cell level, at least not in adults. Or at least I really don't think that's happening at the ganglion cell level. At the amacrine and bipolar level, for sure, I think that's totally happening. And I think maybe a boring example, or at least a simpler example, would be these very wide field amacrine cells. In the mouse retina, we have ones that extend millimeters, and the way that they inhibit cells is by firing action potentials. So they often bring sort of a speedy surround to neurons. In fact, the sidekick 2 ganglion cell, this object motion selective cell, part of the way that it detects motion is by kind of comparing motion in its center with motion across the visual field. And it gets that signal from this really wide range guy. But that guy, or cells like that anyway, also play a role or provide input to just plain old direction selective neurons too, right? There are even older kind of cells with an older definition. They were called Y ganglion cells, Y-type ganglion cells. And they have a very similar kind of huge surround that's extremely fast, sluggish center type thing. And so I think there is a lot of divergence at the amacrine level, and probably at the bipolar level too. Exactly, maybe you're asking, is there a code for that too? Is that sort of pre-specified? Yeah.
Well, that, and I'm also wondering about like interpretation. Like it really goes back to this, the idea with the receptive fields and spots, and with on and off center cells, that the activation of one will inhibit and exaggerate the effect on another if it's neighboring. And so you get these kind of enhanced responses when certain types of cells are next to each other because of the connectivity between lateral, I assume, lateral columns of circuits. I don't know if that made sense.
No, it makes sense, and I think at sort of the interneuron level, it's totally happening, right? Like two neighboring interneurons in your model, driving one, if he inhibits the other, would sort of sharpen the responses on the downstream ganglion cell in the first case. That's like a classical kind of retina pattern, right? That sort of sharpening inhibition. As to whether I know about specific kind of connections, whether, like, let's say the famous Starburst immigrant cells, whether they supply their direction selective inhibition to some other interneuron to influence some other circuit to say that when I see direction selective things, you can't see your thing. Maybe that's what you're asking. That I don't know. I don't think anyone knows that.
Okay.
That would be super cool, but
Yeah, that's what it would be cool if this explains the mechanism, basically. Thanks.
Okay, Tahani has a question.
Thank you so much for the excellent presentation. And I noticed that throughout the presentation today, you focused on the connection between the bipolar cells and the direction selective, the on alpha and the S1. But at one of the few slides you presented at the end of the meeting, you showed, I saw like you were also labeling M2.
like this,
M2 ganglion cells. And I was wondering, because you didn't mention anything about the melanopsin related ganglion cells. So I was wondering if the same connection also applies to non-image forming RGCs, or it's just focused on the image forming RGCs.
Yeah, you copied. I kind of swept that detail under the rug.
Yeah.
I think so, but it's hard to know. So, you know, the phenotypes we saw when we deleted sidekick 1 were strongest on the S1 RGC. We saw some effects on the on direction selective cell. For technical reasons, this cell is hard to label from the brain. And there's reasons for that. Now, we didn't, due to the way that we kind of do our imaging, you know what I mean? We couldn't really separate the responses of this guy from the responses of that guy. They do have awfully similar responses. And M2 cells are for sure intrinsically photosensitive, right? So whether there are deficits here is hard to know. One of the big problems with studying the M2 cell is that it's so low density, right? It's like 15 cells per millimeter squared or something like that. It's like extremely low. Our model would say that it should be doing something. Then again, it has both sidekick 1 and 2. So maybe if you remove 1, 2 steps in to compensate. Maybe the deficit's slightly more subtle. Stay tuned. We're looking into it.
Yeah. Would you suggest that it connect also to bipolar type 2 or type 5, or what? I mean,
what do I think its bipolar connection?
Yeah.
It wouldn't be 2 or 5. It's kind of in the lower part of sublamina 5. It'd be like, I don't know, type 7, type 8, you know, something like that.
Okay.
Would be my guess. We've drawn it here next to the rod bipolar cell, but of course, those neurons famously only connect to amacrines.
Yeah. Okay, thank you.
Yeah.
Philippe has another question.
Yes, sorry. I just I had 2 questions. I forgot the second one. I have a small brain, you know. I was my second question was you know, when the dendrites target specific layers. When you think about retinal ganglion cells, when you think about the axon, I mean, there's aberrant projections all over the place, and then there's a pruning there that is happening. Is that the same phenomenon that is happening, you know, for the dendrites? You have many projections in all directions, and then pruning on the so that.
Yeah, it's a good question. I think in the few cases we know, Philippe, there's an older paper J Neuroscience paper from Josh's group some time ago, where they looked at direction selective cells, the double dendrite ones that I showed you today. Another cell called a JAM-B cell, which just goes to layer 1. Anyway, they looked at a bunch of these neurons, and they looked at them over development. And for the direction selective ones, the classical ones, they seem to be perfect from the start. So they grow straight into layers 2 and 4.
Okay.
The other ones, these JAM-B cells, however, do more of what you're talking about. They kind of grow through the layers, then they refine into the right place. And so maybe the thing to take away is that each type is adopting its own strategy. Some are right from the start. Maybe some take some time to find the correct place. There's a lot of good work in zebrafish on this. I'm trying to remember the guy's name. Jeff Mum, maybe, or someone like this, has done some imaging in zebrafish retina about how their dendrites develop. And I think they find similar things. Some cells are perfect from the start, and some cells refine.
And you believe that that the choice of protein that the dendrites are using in order to achieve specific targeting determines how what the mechanism that is underneath that.
Oh, I see what you mean. Yeah. It's hard to know in the case of the ganglion cell. Okay, but in the case of bipolar cells, I'll make an educated guess. There was some work some time ago from Rachel Wong's lab where I think bipolar cells very early on, when they choose a layer, they do extend these little filopodial things. You know, they go through many layers, and the one she was imaging, I can't remember what it is anymore, does extend these little filopodial processes that that sense a few different layers before they end up in their their adult location. So maybe in our case that would be happening too, although we looked pretty early in the case of type 2s, I believe, and they seem to know exactly where to go right away.
Okay, thanks.
Yeah.
Hideki has a question.
Yeah.
Yes, thank you for your fantastic talk. Related to your question, I'm interested in BC2 and BC5 targeting.
Mm-hmm.
And what is the mechanism of selecting layer by using cadherin? If I look at this phenotype, the layer BC2 targeting layer 2 and the BC5 targeting 4.
Yeah.
And if you look at these, they targeted both. So if I look this phenotype, maybe I think it's a kind of inhibitory effect is abolished. That's why they target layer 2. But thinking about cadherin, it's kind of homophilic interaction. So maybe it make the connection stronger. So I just confused on why this.
Yeah, why do they default to the 2 locations? Yeah. So I suspect, like in the kind of later studies where we went to CAD6 and CAD10 and all that sort of stuff, I suspect it's because they are reacting to the presence of the ganglion cell that's there. There's other factors that prime them to those 2 locations.
Okay.
The choice of 8 and 9 just assigns them to layer 2 or to layer 4.
Mm-hmm.
And we think that it's an assignment because, you know, it's hard to do the misexpression, at least in those days well, but we took a late-born amacrine cell, and we were able to electroporate in constructs to CAD8 or to CAD9. And when we put those proteins in ectopically, we could force those amacrines into those layers.
Okay.
So we really think it's an attractive
okay. So is there any reason to avoid our layer 1, 3, and 5 to target?
Is there any reason to avoid layers 1, 3, and 5?
Yeah.
I don't know about that trio, but there is really nice work from Alex Kolotkin's lab about the role of semaphorins and the boundary between on layers, so the middle of sublamina 3 and downwards, and the off
layers. There's a reversible
Yes, yes. So I think some of these question mark factors that I've put here, I should update the slide, would be things like that. Yeah, sort of a mixture of broad, repulsive, attractive cues kind of set up this layering pattern. Yeah.
Thank you so much.
There's somebody named Bill.
Bill has a question also. Go ahead, Bill.
I'm sorry, actually I typed in a chat box, but nobody looked at it. Sorry. Thank you for the talk. I have two questions mainly. So first of all, why did you choose to study bipolar cells kinetic to layer 2 and 4? Why not other layers combination?
Yeah, it was kind of determined by the expression of the cadherins. That kind of determined the story for us. So it turns out those two bipolar cells, there was a survey going on in the lab looking at a variety of cadherins and IG members and things like that. And then it just turned out that those two cells, the two bipolar cells, one expresses CAD8, the other one expresses CAD9. And we developed reagents to access those neurons. So it began from the expression pattern of the cadherins. Wasn't like a specific, at the time that we chose it, we weren't like, we need to find layer 2 and 4 factors. It was the other way around.
Okay, thank you. My second question is, without the sidekick neuron, SDK, does the connectivity simply drops overall, or is it just the percentage of the correct connection?
Yeah, it's a great question. In the case of sidekick 1, we don't know. In the case of sidekick 2, the ganglion cell that we were studying, I actually did some connectivity mapping, some of that optogenetic mapping to a bunch of other neurons, a bunch of other interneurons that target the same layer, and from the V3 amacrine cell to a bunch of other ganglion cells that target the same layer. And in the absence of sidekick 2, it's not like sidekick 2's absence and the loss of connections that it produces from this VG3 amacrine cell called more synapses from the normally sidekick negative cells. That didn't end up happening. Nor did ectopic synapses from the presynaptic cell form onto these other guys. So it didn't seem like that loss led to the kind of compensatory things that you're thinking about.
Okay, thank you very much.
Yeah.
All right, thank you all for your questions. I'm going to open a breakout room now. So if you want to join, and Arjun, you will have a message coming up and you just join the room. So everyone will have a message, and the one who wants to join us for like more informal discussion or further discussion, you are all welcome to join us.
Sure.